I'm going to switch to English and we're going to start this. And okay, so hi everybody. Our speaker for today is Danna Titelboim from Bristol, University of Bristol. Danna is started off her academic career in the BGU and at Ben-Gurion University in the Negev, did her bachelor, master's, and PhD there, working with Sigal Abramovich, Ahuva Almogi-Lebin, and Barak Herut, mostly focusing in understanding how benthic foraminifera respond to variation in temperature, salinity, and what would future, variations or change in climate and salinity, how would future changes change and affect these ecosystems. She's currently a fellow, is the Royal Society of Khan Fellow at the University of Bristol, and will soon move on to another postdoc at Oxford. She wrote many papers and was very successful in disseminating her research so far, and won pretty much all of the possible prizes at this stage. So it's very impressive. And Danna, we're very happy to hear what you have to say. So take it away.
Yeah, okay, so thank you for that kind introduction, and thank you all for taking the time to listen today. And I'm very excited to tell you about the research that I'm doing here at the University of Bristol. I just want to start by sharing with you something. I have arrived here in February 2020, exactly in time for the pandemic to break. My first message to you today is don't start your postdoc when the global pandemic breaks. Not the best option. And the reason that I bring this up is because a lot of the stuff that I'm going to show has kind of empty, or not empty, but missing parts. And the answer is always because of COVID. But I think we were able to do quite interesting things. And I'm very excited to talk to you today about the research that I've done over this year, dealing with the effect of future warming on benthic foraminifera, and specifically the role as carbonate producers. Okay, so let's dive in. Climate change is projected to impact the marine environment in many ways, from the physiological activity of individuals like growth and reproduction, through species range shift that will ultimately completely change the ecosystem structure. Now, in the case of calcifying organisms, the predicted changes will specifically influence their ability to perform one of their main functions, which is regulating the marine inorganic carbon cycle through carbonate production. Now, nearly half of the carbonate in the oceans is produced in shallow water environments, where, except for affecting the seawater chemistry and regulating it, the carbonate that is produced is also creating very unique habitat that other organisms can then live in. So basically, this has a double function that might be at risk due to warming. And much of the research on the impact of climate change in warm shallow water ecosystem, or at least the most famous part that gets most of the attention, is predominantly focusing on corals. However, there are other groups of carbonate producers that are sometimes being overlooked. So one of these groups, and the one that I would like to focus on today, are benthic foraminifera. And these are unicellular organisms found globally and abundantly in the oceans. And their high diversity allows them to occupy and prosper in a broad range of environments. In fact, they are so abundant that even though they are unicellular small organisms, the carbonate production is larger than 5% of the global carbonate production in the oceans. And their shells actually contribute to the cementation and stability of reef structure. And in some cases, like in low-lying islands in the Pacific, foraminifera are the main source of beach sand, and thus they maintain the stability of shorelines. So this is kind of the motivation to understand the effect that warming will have on not only the well-being of foraminifera, but actually the function or the contribution that they have on the marine environment. Okay, so there are, just in general, three ways or three main approaches to study biotic response to climate change. One is to investigate examples from the geological record. However, it is problematic to make direct comparisons with current and future warming phenomena, as this past ecosystem did not contain the same species and they did not experience the same rates of environmental change as today. Luckily, Foraminifera has modern descents that we can play with in the laboratory. But while laboratory experiments are valuable for isolating the effects of a specific environmental parameter, they are usually short-term, which means that they are excluding any transgenerational processes like adaptation and acclimation that might mitigate some of the effect that climate change will have. And also, they are usually performed on a limited number of species, precluding effects stemming from the complexity of ecosystem interactions, from competition, and from species replacement. So another way to study the effect of warming on foraminifera that kind of maybe combines some of the advantage and disadvantages of geological record and laboratory experiments is provided by a thermally polluted area next to an electrical power plant, like, for example, the one in Hadera that was one of the prime sites that I work on during my PhD. And in this site, the power plant pumps water from the sea to cool the turbines, and then it discharges them back into the sea, creating a thermal anomaly of about 1.5 kilometers along the coast, and basically mimicking future warming under natural conditions. And this is kind of a combination between manipulation experiment done in the laboratory and the natural complexity represented in geological record. So these are the three main approaches. The problem is that observation and conclusions from different studies in this field of how climate change will affect calcifying organisms and foraminifera specifically, they are often show different observations, different conclusions, and sometimes even opposite ones. And this might seem contradicting, but really I think that these are just kind of pieces of a puzzle that we maybe don't quite know how to piece together. And so this is why in my research I try to combine always the three approaches to try to realistically understand some of these complex processes of stress response to thermal conditions. Okay, so hopefully the combination of these approaches allows us to understand the response of benthic foraminifera in a realistic way. And specifically in this study, we want to ask two main questions. And this starts because we know from previous studies that the ability to calcify is reduced under stress, specifically under thermal stress. And this makes sense because calcification is it's a very high energetic demand. And so the reduction or inhibition of calcifications allows a relocation of the energy to more basic physiological activities that are needed for the survivorship of organisms under extreme conditions. Okay, so we know that there will be less carbonate produced, but in this study, the first question that I ask is by how much? We want to try and quantify and understand how much a reduction in calcium carbonate will be by these organisms. And in addition to the decrease in carbonate, it is also important to try and understand how warming will change the material properties of the shells. And this is sometimes overlooked, but it could be important because, for example, if we know that temperature increases the magnesium content, and that in turn affects fundamental properties of the calcite, such as hardness and elasticity, which could affect the ability of the organisms to resist predation and other external stressors. And another example is that if the magnesium calcium ratio is positively correlated with the solubility of calcite in seawater, and we know that it does, this could have a particularly negative effect when combined with increasing ocean acidification. So this is why we maybe want to know more about not only the quantity of carbonate that is produced, but also about the quality. Okay, so starting with the first objective, and this takes us back to the Hadera heat plume, which is really a phenomenal site to study changes in shells under natural conditions. And we could also avoid making all kinds of assumptions that we might want to make when trying to make this assessment, because we already had quite a lot of information about this site and about the foraminifera that lives there, because of the two first chapters of my PhD. So I just want to very briefly talk about these chapters, as they are basically the fundamental assumptions of this project. And I will try not to get too much into it, but hopefully just give you enough background. Okay, so what we did, we monitored 2 stations in the thermally polluted area. The first, marked in red, we call it H2, is at the core of the plume, very close to the hot water discharge. And the second, marked here in green, we call it H4, is at the edge of the plume where temperatures are only slightly elevated. And we monitored an additional control station about 18 kilometers north to the power plant, so it cannot be affected by the discharge of warm water. So we monitored the environmental condition and the foraminiferal population at these 3 stations for a duration of about 15 months. And the temperature were monitored in a very high resolution, as you can see here in this graph here. And you can basically see that at the center of the plume, marked in red, temperature is much higher than the control that is marked in blue. And we're talking about an average of about 5 degrees increase and up to 9 degrees. While at the edge of the plume, marked here in green, the temperature are only slightly higher than the control. And we're talking about an average of 1.5 degrees. That's all. So that's very close in the future. Basically, the heat plume is a natural laboratory, as I said before, and the 2 stations that we have chosen are kind of a simulation of the near future warming at the edge of the plume and then further into the future at the core of the plume. Okay, sorry. So monitoring of the foraminiferal population showed that the local species at the Eastern Mediterranean can be divided into 3 groups according to their thermal tolerance. And the first group, marked here in blue, are species that occupy the control station but do not inhabit even the edge of the plume. This indicates that they are very close to their thermal tolerance, and with even a small increase in temperature of only 1.5 degrees, they will no longer be able to exist in this area and they will have to migrate away. They cannot survive here. But in our context of changes in carbonate production, this basically means that 100% of the production will be eliminated. So no further study is needed on these species. And now moving to the second group, these are 2 species that are resilient enough to survive the 1.5 degrees increase, but too sensitive to live at the core of the heat plume itself. So they are kind of semi-sensitive or semi-resilient. And in this group, there are, as I said, 2 species, but one of them, this one is an agglutinated species. This means that it basically picks up particles from the environment, so sand grains, and kind of glues it to its shell. So his shell is less about carbonate, although it does produce some carbonate, but not a lot. Mainly it's just agglutinated parts. And so it is less interesting in the context of carbonate production. However, the second species, this one, is called Physiogenic, and it's actually very important calcifier as it is the main component of sediments in many geographical locations. And specifically, for example, in the coast of Israel, in many locations, this species is representing over 90% of the sediment. And in fact, this species was sometimes so, in the geological record even, was so abundant that it was referred to as living sands. Okay, so this will definitely be the first species that we focus on. And now moving on to the third group, and these are 4 species who are resilient enough to live inside the plume, indicating the very high resilience as they can survive this extreme warmth in this station. So focusing on these resilient species, and in the graphs here, you can see the numerical abundance in the red bars and the monthly average temperature in the black dots line. So you can see that out of the 4 species, only 2 are present consistently throughout the year, even in the extreme summer months, which makes them really good candidate for our study, as there obviously will still be players in a warmer world scenarios. And interestingly, Paravotalia show a decrease in abundances between June and July, when temperature is around 34 degrees, much higher than all the other species that decrease their abundances between April and May, when temperature is much lower, around 30 degrees. And this is the first indication that Paravotalia might be an extremely resilient species and definitely more resilient than the other species that inhabit the plume. And then the next step of that study was to examine whether the 2 resilient species that occupy the plume throughout the year, do they actually calcify under these very high temperatures, or do they just survive there? So we used the shells collected throughout the monitoring, throughout the 15 months of monitoring, and we used magnesium calcium thermometry on the less chambers of the species to determine the calcification temperature of the last chamber. So basically, we tried to find the extreme, the edges of temperature that these species can calcify in. And we saw that both species are not limited by the high temperature of the plume. However, they are not calcifying in the colder part of the year. And I don't want to go into details of that because that's a whole other lecture. But what I want to show here in this part is that even though these shells were collected from the same place and at the same time, which means that they were exposed to the same range of temperatures, Pararotalia, the species marked in pink, calcified under higher temperatures compared to the other species, Lachlanella, that is marked in green. So we have 4 species, but only 2 of them actually occupy the plume throughout the year in a consistent manner. And we have different thermal tolerance between them, with Pararotalia being more resilient than Lachlanella. Okay, so we now know what species to focus on thinking about carbonate production in a warmer world. And not only are these the major players, they also represent 3 levels of thermal resilience. Pararotalia being the most resilient species, then Lachlanella, and then Physiogenic, which again can tolerate 1.5 degrees, but not much more than that. Okay. So what did we do to estimate the changes in carbonate production by these species? We took shells that we collected throughout the monitoring from different months throughout the year, trying to cover every other month, and we CT scanned them. Basically, when you CT scan something, you produce hundreds of pictures of 2D pictures that you can then stack together to create a 3D model. And here it's just a simplified example for each of the species, but you can see that the species are very different from each other, but more importantly, you can see the complex internal structure of each of the species that allows us to build a very accurate 3D model that we can then perform measurements on. Okay, so these are just measurements of 2 parameters of the morphology of the 3 species, the size and the shell thickness. And these are all measurements from samples collected from the control station, from Nachsholim. So this is basically their normal measurements for today, and this represents the full year. And the reason that I show these 2 parameters is that I think that these are the parameters that are most important in terms of morphological changes causing changes in the amount of carbonate that an individual can produce. And basically the 2 options are that either the foraminifera gets smaller, so it reduces the amount of carbonate because it's smaller, or it doesn't change the size, but it makes thinner shells. So that's also a way to reduce the amount of carbonate that you make. And of course, it can be any sort of a combination of the 2, all the way up to perhaps smaller, thinner shells. And these are all things that we see in different geological records. So we want to try and also understand why we see different things in different records of supposedly the same kind of climate change. Okay, and the last thing that I want you to notice in this slide is that the species are very different from each other. And the reason that I mention it is just to highlight that you can really only compare one species to itself. So for example, if this species, Physiogenic, which is obviously much larger than the other 2 species, if it reduces the size even by quite a lot, it will still be much larger than the 2 other species. So you have to make sure that you compare kind of apples to apples and not make a fruit salad out of it and start comparing different things, which might sometimes, or it might be more difficult in a geological setting where you don't actually know what is a specific species and what are similar ones. Okay. So let's start to look at the results about changes in carbonate production of the species, and starting with the most resilient species. The upper panel shows the abundance in each of the stations throughout the year, and this is the color line. And the black dots are just a daily average temperature that we measured in the station. This is all data from my PhD still, and I just put it here because you can really see that there is a decrease in abundance between the control station and the core of the anomaly, and we're talking about a decrease in abundance of about 70%, sorry. So this already means that there is a lot less carbonate because you have less individuals, then you have less carbonate shells. So now let's look at the left graph here, and this shows size in respect to time. And we can see the seasonal cycle of size, and we can see basically something very different between the stations. In the warm station marked in red, we can see that there is one reproduction event, and in the control station in blue, we can see two reproduction events. And the reproduction events are represented here by the dotted lines, that they show the transition from moving from larger shells to smaller shells. And the only way that foraminifera can become smaller is if they reproduce and start another generation, because foraminifera shell is not going to just become smaller unless there's dissolution, and that's not relevant in the setting that we're working in. Okay, so we see a change in the reproduction and change in the seasonal cycle, which can then possibly explain the change in the abundances. But also when we look at the graph here, we can see that the size doesn't really change, even though it reproduces only once a year, it doesn't become bigger or smaller, it retains the size. However, when we look at the shell thickness, we see that it's kind of similar, but there is a slight shift to thinner shells. And this becomes more clear when you think about it as annual averages. So if you think about it as annual averages, there is a significant decrease of about 10% in shell thickness of this species. So moving to the second species, slightly less resilient, we're talking about the Lachlanella, the one that you can see here. Again, you can see the abundances between the station. You can again see the quite dramatic decrease in abundances. However, now when you look at the seasonal cycle here, you can see that there are two reproduction events of each of the stations, but they are slightly shifted in terms of when they're happening, but it's a very similar seasonal cycle. However, it's also very clear that the shells are much smaller. And I don't think this needs explaining, because you can just see that the blue dots are much higher than the red dots. So specimens in the control station are much bigger than in the warm station. You can see it in both graphs. So no change in reproduction, however, much smaller shells. And if we look at the shell thickness, then we basically see the same values, no change in shell thickness. And finally, looking at the more sensitive species, the Physiogenic. And again, looking at the abundances, I just remind you, it does not exist at the core of the plume, just at the edge, indicating its kind of sensitivity. And you can see that there's a very dramatic change. So obviously a lot less carbonate is being produced, because it's like a 90% decrease in abundances. And also, it has a slightly different seasonal cycle. So these graphs are a lot less details, because this is still a work in progress. And on this species is working one of my MSc students. So it's hard to say anything about the seasonal cycle, but you can definitely already see that the specimens are decreasing in size. So there are a lot less of them, and they are becoming smaller. And for such an important calcifiers, as I said, sometimes representing 90% of the sands in different settings, this could be a major decrease in carbonate production of these species, with even just a slight warming of about 1.5 degrees. Okay, so just to wrap this part up, basically we have 3 species with 3 different thermal tolerances, and each one of them is doing something different. The most resilient species retains the size, but changes the reproduction cycle, and also produces slightly thinner shells. The middle species, the one with slightly, still very resilient, but slightly less than Pararotalia, produces much smaller shells. And then the Physiogenic produces smaller shells, but I'm not putting any quantitative values on that until we actually finish quantifying it. And okay, this allows us to kind of maybe try and understand what will happen to carbonate production by benthic forams in the Eastern Mediterranean. But this also brings up the hypothesis that actually carbonate reduction of individual foraminifera happens in 2 stages. First, when thermal stress begins by decreasing the amount of carbonate by producing thinner shells. And then as stress increases and becomes more acute, then they reduce the size of the shell to reduce their calcification to save the energy for other purposes, more basic for survival. And okay, that's a good hypothesis, but that doesn't really, it's not really proven from this data, it's just an idea. So how can we check that? How can we examine that? So yeah, we move to the geological record. And our example from the geological record is from the Paleocene-Eocene thermal maxima, about 56 million years ago. And this is a rapid global warming event with an average increase of about 5 degrees. And because it is relatively rapid, it is often used as an analog for current warming. And when I say relatively, I mean relatively compared to other geological examples, not to today's warming, which is obviously much faster. But it is still considered a good analog and is used quite often to study the effect of climate change. And what you can see here is a plot and a text from this paper here that was published in geology by Stuart Robinson from Oxford. And it's really interesting because he worked on an ODP core of a ground carbonate platform in the Pacific. And he did very high resolution chemostratigraphy. So basically he did the delta C13 to find exactly the boundary, the PETM, and in his own words from the paper, to permit an initial assessment of biotic and environmental response of shallow water lagoon. And I thought that this is actually a really good platform. So he have done, so the stratigraphy is done by the ODP scientist, and now he has done this high resolution chemostratigraphy. So we know exactly where the boundaries, and we know kind of the rates of the warming and the cooling. So this is a really great platform to try and investigate first changes in carbonate production in geologic record, and specifically our hypothesis from the modern species. So the plan is basically to scan the rock samples from this core and then look at the individual foraminifera shells within each of the rocks, segment them, and do the exact same measurements that we did on the modern species. So I did order the core, and it took a long time because of COVID, but I did get it very recently. And I don't have quantitative data yet, but I still wanted to show you what I've done so far because I wanted to see the amazing potential that this core actually has. So I'm just going to show you some of the scans, just going upwards with the warming. And starting about 56.6 million years ago, we see a foraminifera waxstone. So definitely some biogenic components of small miliolids and orbitolinids. Doesn't really matter, small foraminifera in this rock. Moving upward, just before the onset of the warming, estimated at 55.4 million years, we see a very similar thing, waxstone with small foraminifera. Then about halfway through the warming event, we see a major change with additional organisms like gastropod and bivalves, and again, smaller miliolids. So we definitely see a biotic response to the warming, a positive one. But then at the peak of the warming, at the warmest part of this core, we see nothing. We see a mudstone with no fossils at all. Then immediately after, as cooling begins, we see again high diversity with large and small foraminifera, as well as coralline algae. So we see quite a quick recovery of the ecosystem. Then about 500,000 years after that, we still see a biogenic rock, but with less diversity and dominated by a large foraminifera. And then after around additional 3 million years, with more cooling, we see small agglutinated and small miliolid forams in this waxstone. So this is our tour of the PETM in the ODP core. And I think this is remarkable because this really shows the entire process from before the warming, through the warming, and the entire the entire recovery. And by the way, this core is going further. So the entire recovery process. And this really presents a wonderful example of the biotic response to climate change. So the main problem that I had when I started working on that is that I thought that I will randomly choose specimens from this rock to perform the analysis that I did on the modern specimen. And really, there are so many specimens, there's such great variability. It's much harder to identify species in a rock compared to real modern that you can take them and look in the microscope and start turning them around in the rock. It's really much harder to do that. So obviously you need a much larger sample size to create any robust observations. So we will use methods including machine learning to segment the different components in each of the sample to create large data sets and And so the next stage is finishing that up, and this will help us achieve two purposes. The first is to examine the hypothesis about the way that foraminifera reduce their calcite, first by thinning and then by dwarfism. And the assumption is that you can see it with the warming process. We can see the thinning and then the dwarfism, but we'll see what we see. And the other thing that came up is that this actually presents an amazing opportunity to quantify the changes in carbonate by all players in this game throughout the event. And once you start using tools like machine learning, and you can really train the software to know what this gastropod look like, bivalves look like, what this foraminifera and other foraminifera, and you can train it to recognize these shapes, then you can really create a very robust, big quantitative data set to show how carbonate production has changed during this event of rapid warming. Okay, so now that we know locally what will happen with carbonate production of Benthic Foraminifera, and we have a plan to how to test this hypothesis and how to use this in larger scales, we can move to the second part of my research that examines changes in material properties of the calcite shells. So to do that, we use Raman spectroscopy that provides basically indication of the chemical composition and the crystal structure of minerals. And this provides an efficient and cost-effective way to identify changes in material properties. So here you can see the Raman spectra of a foraminifera shell. And I'll just walk you through what you see here, because this is like the basis of all the results that I'm going to show you. And okay, so Raman spectra of calcite has four main modes, four main peaks, two external modes that change according to the vibration of the carbonate ion to the calcium or magnesium ion. And this is marked in red. So there's a transitional and librational vibration, and changes in these vibrations will change basically the location of this peak to higher or lower wave numbers. And I just want to say about this small peak. So this is this peak, and this small peak here, I have to tell you, I don't really know what it is. It's not actually a part of classic calcite. There is some indication in the literature that this might be related to non-traditional classification mechanism. In any case, this is not, I'm not going to talk about it right now because I'm not sure exactly what it means. So in the context, it's not that important, but it's there. So I'm showing it, but I'm not going to address it in the results. Then we have the two internal vibration, the two internal modes, internal peaks that are related to they're called internal because they're related to the vibration within the carbonate ion. And I'm going to be honest here and tell you that this peak right here, the in-plane bending, we can't actually measure it that well. So I'm also ignoring that when I look at the results, because I only want to look at things that are very solid measurements. And the other three peaks that you see, this one and these two, are basically produced by calibration labs that we put in the system. So we can use this peak to calibrate the machine and have a very high accuracy measurements of the calcite that we measure. So basically, to sum that up, we are going to look at three peaks: the internal peak, representing the symmetric stretching of the carbonate ion, and then the two external peaks representing the librational and transitional vibrations between the carbonate ion and the calcium magnesium ion. Okay, so I want to start by talking about a study that was published in 2017 that shows really quite in a wonderful way how the position of the Raman peaks, they shift to higher wave number as magnesium content is increasing. And they use multiple organisms with all kinds of magnesium concentrations. And here you can see the main figure from that showing that indeed in three of the peaks, but they show it also for the fourth one, there is an increase in the wave number with the increase of magnesium. So that is pretty cool. However, you can see that this data is kind of messy. And I think it's pretty reasonable that it's pretty messy because there are a lot of organisms involved here, and magnesium is not the only thing that is changing the Raman spectra. So I took the liberty to replot some of their data, and I've plotted the internal mode against the magnesium measurement that they have, and I just colored the data according to the organism that it was measured from, and you can see really that there is quite a nice separation between species, the algae, the ectoderm, the barnacle, and so on, and just the foraminifera are kind of everywhere. So I now colored just the foraminifera. I turned all the other organisms to gray, and I just colored the foraminifera, and we can see that they actually still split into groups. So perhaps there is a specific Raman spectra for different foraminifera, and this could be related to two things. So first, you can see it on the graph, it can be related to magnesium. There are low magnesium and there are high magnesium, and they're very nicely separated. However, here there isn't such a separation in magnesium, and still there is a separation in the wave number. And also, other than the fact that these have different magnesium values, magnesium content in the shell, these are also very different kind of shells, and I'll talk about it in a second. But these are hyaline species, these are mediolite species, so different calcite, different Raman spectra. Why not? So basically, what happened when I finished staring at the data from this nice paper was that I realized that if I want to work on the Raman spectra of foraminifera to try and identify changes caused by thermal stress, I really need to understand what makes these differences. Is it a species thing? Is it an environmental thing? Is it a shell type thing? What? So the first thing that I did was to take different species and measure them. And basically what you see here are the 6 most common species in the Eastern Mediterranean. And all of the shells that you see that are part of this data set were collected at the same time from the same place. In this case, it's from Nachshalim in the northern coast of Israel during July. The reason that it's during July is that this is the time of year that pretty much all of these species are not juvenile, but they're also not fully grown. So there is a good chance that they are still growing, that they actually calcified around the time that we picked them up. And this is not to say that they've calcified under the exact same condition. And we saw that before with Paraotalia and Lachlanella, that even though that they're collected together, they have very different magnesium calcium, very different classification temperature. The same thing is probably true here, but this is the most confinement that we can get. These are species who calcified under the same conditions or in the same range of species. And we include here the 2 shell types, hyaline and mediolite, and just a short reminder for the non-foraminifera people among us. And this is a model from a paper published in 2019 showing the different classification mechanism producing these different type of shells. Basically, mediolite species, they produce calcite needles intercellular and then kind of pushes them out, while hyaline classification is external, is external to the cell on an organic template. Basically, the one thing that you really need to understand is that these are 2 different mechanisms producing different kind of calcite. Basically, mediolite are calcite needles arranged randomly in dense rows, while hyaline are multi-layered radial calcite, creating kind of a glassy appearances. And that's in contrast to the mediolite that have kind of an opaque white appearance. So different kind of calcite that could cause different Raman spectra in theory. So this is why I have mediolite species and hyaline species here. And the second thing, or the obvious thing that could change, is the magnesium concentration. And again, I wish I had, the actual magnesium concentration of these shells. However, COVID. So hopefully I will have them soon enough. For now, I just rely on what I know from the literature about each of the species. And I know that all the mediolites have high magnesium. This is not to say that they have the same magnesium contents, but that it's in the same ranges. And this is in contrast to the hyaline that have very different magnesium content. We have very low magnesium shell, and we have high magnesium shell, and we also have an intermediate form. So this is this is just to cover all the options of what I wanted to see here. And now we can compare the changes or the differences in the 3 Raman peaks that we are looking at between all these different species. So let's start with the internal peak. It's this one. And first of all, we can see that it might have something to do with the magnesium content. It's kind of hard to say because we don't have the actual magnesium concentration, but we can see that there is definitely the low magnesium has lower wave number. And then between the intermediate and the high, it's hard to say because of the large variability of between the specimens. So this will need further, but it doesn't really make a lot of sense. So it doesn't really make a lot of difference. We know that there is a change, a general change with magnesium, whether they're different or not. Some of it is definitely similar. And so now comparing between hyaline and miliolids, we see that the high magnesium hyaline and the high magnesium miliolid shell, they are similar to one another. So it's definitely not a shell type thing, because otherwise they might be different. Okay, so this is that. And now we look at the external peaks and we see something different. We see that the low magnesium is very different from all the other species, but all the other species, including the intermediate and all of the high magnesium, they look completely the same. They look very, very similar. And so they're definitely comparable. Okay, so basically what we know now, we know that the Raman spectra probably doesn't change due to shell type, and it probably does change with the magnesium content, but it's only significant when you look at very low magnesium to the other groups. Okay, so we are now back to focusing on our three species, the one that we looked at before, and we have the Physiogenic that is relatively sensitive, and we have the Lachlanella and the Pararotalia from the plume. And again, we can see that there might be some differences between the intermediate and the high magnesium in this peak, but the two external peaks, they look exactly the same for all three species. And now that I feel rather confident that I understand what can be compared and what cannot be compared, I can move on. I just want to mention this again. I do need the chemical data of these specific shells, but I'm pretty confident that it's going to validate what I'm saying and not dispute it. But I just want to put it out there that this is not a closed case. Okay, so now that we know all of that, we want to know basically what does temperature do to the Raman spectra of Foraminifera. And we thought that the ideal way to do it is basically to culture them in a lab and then measure the calcite that they produce. So this is what we did. We took shells of Physiogenic and we cultured it in a lab under different temperatures. And I just want to say that the reason that we use Physiogenic is a practical reason. It's relatively easy to culture it, and it produces the shell in quite a rapid way in comparison to other species. So the way that this works is that before we start the culturing experiment, we actually culture a bunch of these guys in a fluorescent stain, which creates fluorescent calcite, and you can see this in the part here. And then when we start the experiment, we actually move them to natural seawater, so then they continue to calcify non-fluorescent calcite. The reason that this is important is because after the experiment, when we take them out of the experiment, we can actually see this indication, and we know that this is exactly the time that we started the experiment. That all of this calcite, if we measure whatever chemical Raman, whatever properties that we want, this is calcite that was produced under the conditions that we wanted. Okay, so what did we do? We cultured them in temperature between 15 and 35 degrees. This basically covers the entire temperature that they're exposed to in the Eastern Mediterranean, and including also the warming scenarios. And you can see by the kind of parabolic shape that we actually cover the entire range that is relevant for this species. We see that in 15 and in 35 degrees, there's hardly any calcification. There's no inhibition of calcification. So that's very interesting, but really nothing to measure there because there's no calcite. So in this study, we focus on the range between 20 degrees and 32, and we measured calcification rate in each of the weeks. And the reason that we do it is because calcification rate is actually an indication for the well-being of the Foraminifera. And what you see here is that there is some sort of optimum in 20 and 25 degrees, and then there is a mild decrease in 30 degrees, and then a major decrease in 32 degrees, indicating that this is a calcification already done under probably stress response. Okay, so we took the shells from all of these, like literally the same shells from these treatments, and we measured the new calcite. And we saw that basically 25 calcite that was produced under 20, 25, and 30 degrees showed the same position of peaks, of the three Raman peaks. And only the several specimens that we culture under 32 degrees, they show a very different Raman spectra. They show a shift to lower wave numbers of each of the peaks. And this is done in a very drastic way. So this is most likely stress response, because if this was the effect of temperature, then we will see a gradual increase. That would make sense because there is an increase in magnesium and so an increase in the wave number. But this is not what we see. We see not a continuous change, but an abrupt change to lower values. So this is why we feel like this is actually an indication for stress response, and we were really, really excited about that, because this actually means that we can see that we have a proxy for physiological thermal stress. And so we can maybe use that to see things in the field. So we wanted to further examine that. So we said, okay, let's take this and see what we can see about these shells that we collected from the thermal pollution, from the thermal anomaly site. And so I just want to remind you something that I talked about in the beginning, but just so everyone remembers, we're talking about these two species. That we collected them from the core of the plume and from a control station. And when we did magnesium calcium thermometry on their last chamber, we saw that they calcify at the high temperature of the plume, but not at the cold ones. They are limited by the cold winter in the Eastern Mediterranean. And further to that, they have kind of different calcification temperature, where Parvotalia actually has a higher calcification temperature. And I didn't stop on that, but I'm just going to say it. They have higher calcification temperature, and they also have higher magnesium values because of that calcification temperature. Okay, so what I did was to take shells that were collected during the monitoring, and I took them for 4 kinds of samples representing 4 thermal conditions. The first is a group of shells that I took from warm conditions. This is basically the summer, like the most extreme summer months in the warm station. Then there is the normal group. This is just spring in Naftolim, basically just normal temperature, ideal, go to the beach, this is fun. And then we have a cold month, which is the winter, but in the warm station. And then we have a very cold month, which is the winter in the control station. And as I said, and as we saw from the magnesium measurements, basically this was too cold for either of these species to calcify. And this is important because this means that they didn't calcify when we picked them up, but the last time that they calcified was actually like the coldest temperature that they could calcify in. So we were pretty optimistic that we could see stress from that. Okay, so basically the x-axis here in the top graph is also time, and this is the change in this Raman mode with time. But it was actually more interesting to plot it against magnesium. And it was a little surprising that we actually saw this linear correlation. And it was surprising because we did not see this correlation when we were looking at the Physiogenic culture in the lab. This could be related to the fact that this is an intermediate magnesium and this is a high magnesium. So the scale of change in magnesium is different. I'm not quite sure yet. We'll come back to that. However, we can see that there is a linear correlation between the position of the Raman mode and the magnesium values. And the reason that I'm quite confident about the reliability of this regression is that it has the same slope as non-biological carbonate and as biological carbonate, as was shown in this paper from 2017. So same slope, this makes sense that this is reliable, and we can see that when we take the 4 thermal conditions with the 2 species, we actually see this beautiful linear regression, except for 2 cases. So there were 2 cases where the samples actually fall off the linear correlation. And let's look at the first one. It's this one. It's the one from the warm station. Basically, we see that even though magnesium was high, it has a lower position of the Raman mode than expected. And this is, again, exactly what we saw in the Physiogenic experiment. We saw a lower wave number with thermal stress. So we see that here too. However, surprisingly, we also have this point. This is the one from the thermal condition that I call it too cold. It's too cold to calcify. So basically cold stress. And you can see that this also deviates from the correlation, but this time to a higher wave number compared to what we would expect considering the magnesium calcium. Yeah, so this was quite surprising, but we are pretty confident that what we see here is stress response and possibly a warm stress response and a cold stress response. And this is what we think that we see here. And I just want to put here also the external modes, because we saw something interesting. First of all, you see that the position of these two modes are different, very different, significantly different between Parautalia and Lachlanella. But more importantly than that, we can see that Lachlanella, the three groups are similar to each other. However, the one from the two cold condition is very different from them. So considering all of this together, this is probably the indication of stress during calcification. Yeah, okay, so I'm really running out of time, but I just want to conclude this. I'm very excited about this. I think there is really great potential to use Raman spectroscopy together with traditional proxies such as magnesium calcium to actually see stress response in the record. Of course, this still needs work. And the very next step is to do the same thing that we did with the Amphithygina in the lab, but this time to these two species cultured in the lab to see what kind of correlations they produce. And we also have a plan to examine this via collaboration with people who culture other organisms to see if this thermal stress response is also occurring with other organisms. Okay, so I'm not going to read through this because I didn't think that this would take so long. Basically, take-home message, the integration of laboratory and field experiment is really valuable. And I'm sure that once we progress with the geological record, it will even make more sense. Okay, I was going to talk a little bit about what's going on next with me. So I'm just going to say it very briefly. In a little less than 3 months, I am finishing early my fellowship here to start my Marie Curie fellowship in Oxford University on a project that I'm extremely excited about. Because if you remember, I talked before about the problem with laboratory experiments is that it excludes transgenerational mitigation that could happen over a very long process and many generations. And we're going to focus on two groups, coccolithophores for open ocean and large Benthic Foraminifera for small ocean. We're going to do experiments of several generations and see what happened to calcification and photosynthesis in order to try and understand what kind of biogeochemical implications the adaptation of these organisms have on the chemistry of the ocean, and potentially what interaction that has with shallow and deep waters. Okay, I really thank you for your attention. I also want to thank the funding that I got to take these fellowships. Thank you.
All right, thank you so much, Danna. It was a really great talk. We have time for a couple of questions. So if you have a question, unmute with yourself and ask away.
Oops. Sorry, there was some problem. I couldn't hear that.
Sorry, Danna?
I couldn't hear you. I had something when I pressed, I don't know.
Okay, that's okay. Okay, I have a question, and then maybe others will follow. So do you have any hypothesis that explains what physically or chemically is changing in the calcite crystallitis between that is type related to type and not to magnesium content, right?
Sorry, did you ask about the different shell types?
Yes, I mean it's calcite, right?
It's calcite.
And Raman, you know, is basically you're looking at the vibrational modes of atoms and bonds in the crystal lattice.
Okay,
so if you're not changing magnesium content, what is different then?
Okay, so that's a very interesting point. There's a couple of things about that. So magnesium content, we know that it makes a difference. I just want to say something about Raman and calcite. It's quite common in kind of earth science to use Raman to see differences between minerals. And this is not exactly what we do. We want to see differences within calcites. And the real question is, you know, how accurate can you be to see really small differences in these vibrations in the molecules? And the answer is that you can be very, very accurate. And this is done in material properties all the time. So we can actually see differences between calcite, and every small difference you can theoretically see it. And we don't see a difference between the shell types, which is strange because you would think that a calcite that actually visually looks so different would do something different, but apparently it doesn't. Other differences and this is what we think that we see as thermal stress, is that we think that as stress accumulates, the organism has less control on the creation of the calcite, and so it creates more defects in the lattice. And so this is basically the change that we see in the Raman. Okay. But we don't actually know that. This is just an hypothesis, and the way that we aim to check it is to start checking the actual material properties. The main problem with that is this is kind of an expensive analysis, and I'm funded by the Royal Society with a very generous fellowship, but it's not quite enough to do everything. So I did ask them for some more money, and this is the only time that I can say thank you, COVID, because of COVID they actually agreed to give it to me. So we're going to do some atomic force microscopy, some nanoindation to try and see the actual material properties, to try to see what does these changes in Raman spectra actually show.
All right, thanks. Do you have more questions?
I have a question.
Go ahead.
In the plume in Hadera, are the changes along the plume only temperature, or are there any modifications of the carbonate system, like pH or carbonate ion supersaturation change?
Okay, so I will give the simple answer is that there are no changes, but I'll give also the more elaborate answer is that it's possibly related to the resolution. So we did very high resolution of the monitoring of temperature, but less for the other environmental conditions. So we did, every time that we sampled, we measured the pH and other environmental parameters, but we don't know what happened continuously throughout this time. I mean, it could be a good idea to push a pH logger at this station to figure it out. We don't think that there is any significant difference in this environment, but just to be completely fair, we don't know.
Thank you.
Do we have time for one more question? One. Okay, I think you're off the hook. We're done. Okay, so I'd like to thank you again for giving us a great talk, and thank everybody for joining us. And we're going to meet again next week in person. The next seminar is going to be actually in classroom 109. So we'll see you there next week, most of you at least. Bye, guys.
Bye-bye.